Hello and welcome back to another session of the Deep Dive. Today, we are not just, reading through a textbook. We're trying to decode the machinery that actually keeps us alive.
That's a great way to put it.
I want to start by asking you to do something very simple. Just look at your own wrist. Or, if you can't see your veins clearly, just picture a sample of human blood.
It's a simple image, I think, but there is so much chemistry hiding right there in plain sight.
Exactly. We all know blood is red. We learned that in primary school. But here's the real puzzle. Why is the oxygen-rich blood, the stuff leaving your lungs, a bright, vibrant, scarlet red? But the blood coming back from your tired muscles, the deoxygenated blood, is a sort of dark, moody purple or blue. It's the same liquid, isn't it?
It is the same liquid, the same cells flowing through the same person.
So why does the color change so drastically? Just because, the oxygen has left the building?
It is a fascinating question, and the answer, believe it or not, isn't really biology. It's chemistry.
It's chemistry.
Specifically, it's the chemistry we're discussing today. Coordination compounds.
That brings us to our topic. Importance and applications of coordination compounds. This is from Unit 5. Now, I know what many Class 12 students are probably thinking right now.
Oh, I can guess.
They see this chapter, they see those square brackets, complex naming rules, oxidation states, and they think, oh God, this is just another theoretical topic I have to memorize for the board exams.
I completely understand that feeling. When you're just staring at a page full of diagrams, it feels like abstract geometry, not, real science.
Precisely. So our mission today is to prove that thinking wrong. Why should a student actually care about this topic beyond just getting the marks?
Well, we should care because if you take away coordination compounds, you basically turn off the lights on modern life. The source material uses a very strong phrase here. It says, these compounds are the inhabitants of a vast ecosystem.
An ecosystem. That sounds more like biology than chemistry.
It's a metaphor, but it's a very accurate one. These compounds are, they are everywhere. They are in the hemoglobin in the blood we just talked about.
Right.
They're the drugs like cisplatin used in cancer treatments. They are the catalyst in factories used to clean dirty water or even make plastics. So understanding this Unit 5 is really the only way to see chemistry as a practical science that, solves real-world problems.
So let's unpack that, this idea of an ecosystem. At the simplest level, how should a student visualize these compounds?
At the simplest level, don't think of them as just drawings. Think of them as tools. Imagine the whole chemical world is a massive construction site or maybe a busy factory. Coordination compounds are the specific machines doing all the heavy lifting.
Okay, so they're the machines, but what's the engine inside these machines? What is really happening at the particle level?
The engine is the metal ligand complex.
Yeah.
This is the core concept of the whole chapter. You have a central metal ion like iron or copper or platinum, and it's surrounded by molecules called ligands. This setup, the structure, allows them to do very specific jobs.
Specific jobs like what? Can you give us a quick overview?
Certainly. Let's look at three main areas. In biology, we have enzymes. These are basically huge protein molecules with a tiny metal ion like zinc or iron just sitting in the middle. And that metal ion helps catalyze the reactions that keep you alive. Then in medicine, we have these platinum complexes designed to, hunt down and kill cancer cells.
Wow.
And in industry, we use them to facilitate reactions, like making ammonia for fertilizers or polymers for plastics.
So without that central metal ligand bond, none of this works.
None of it. The biology stops, the medicine fails, and the factory, it shuts down. That bond is the functional part of the whole machine.
And that puts a lot of weight on those coordinate bonds.
Yeah.
Now, for the students listening who are thinking, okay, I get the concept, but I need to write a proper definition in the exam, we have an anchor definition from our source. This is the one that's safe for your exams.
Yes, this is very important. If you have a notebook, please take this down. I'll read it slowly. Coordination compounds have extensive applications across biology, medicine, industry, and environmental science due to their ability to selectively bind metals, facilitate catalysis, transport molecules, and produce specific colors and magnetic properties.
That is a very dense sentence. It covers a lot of ground.
It does, but that's why it's so good for the exam. It mentions the fields biology, medicine, industry, and it mentions the functions binding, catalysis, transport, color. If you write that down, the examiner knows you understand the full scope of this chapter.
That's a great tip. This brings us to what the source calls the trigger and fix. The trigger is that common frustration students feel.
The why do I have to learn this abstract nonsense feeling.
Exactly. Students often think coordination chemistry is just, about drawing shapes or memorizing IUPAC names.
And the fix is to realize that these are the inhabitants of the ecosystem we actually live in. I mean, we are literally made of coordination compounds. You're not just studying lines on a paper, you're studying the machinery of your own body.
To help us understand how these tools work, the source material uses a brilliant analogy. It compares a coordination compound to a molecular Swiss Army knife.
I love this one. It makes the concept so concrete. Think about a Swiss Army knife. It's a single object, correct?
Right, it's that red pocket knife with all the little gadgets.
Exactly. You pull out one side, it's a blade. You pull out another, it's a pair of scissors. Another is a bottle opener. It's one single tool, but it has multiple functions packed right into it.
So in this analogy, the coordination complex is the knife handle itself.
Sort of. Think of the central metal ion as the handle. And the ligands, the things attached to it, are the specific tools. So depending on which ligands you attach to that metal, the complex can do different things.
What kind of different thing?
Well, one setup might be for binding specific metals. That's what we call selectivity. Another setup might be for speeding up a reaction. That's catalysis. And another might just be there to absorb light and produce a beautiful color, which is used in pigments.
A multifunctional toolkit.
Yeah.
There's another analogy mentioned briefly, the keys and locks analogy.
Yes. This one is crucial for understanding how they work in medicine. You can think of biological systems like enzymes or receptors in your body as very specialized locks.
And the coordination compound is the key.
Correct. But it has to be the right key. The ligands, they act like the ridges and grooves on the key. Only very specific ligands will fit into that biological lock. This is why we can design medicines that target a specific disease without ruining the rest of the body.
That's the concept of selectivity again, right?
Precisely. You don't want a medicine that just binds to everything in your body. That would be a poison. You want it to fit only the specific lock it's meant for.
That really explains why they're so useful in medicine. But let me ask a broader question. Why does this concept even exist in our syllabus? What problem did it solve for science?
It exists because we simply cannot explain the world without it. Before Alfred Werner proposed his theory back in the early 1900s, chemists were really confused. They couldn't explain how hemoglobin carried oxygen. They had no idea why some compounds were brightly colored while others were just, white.
So before this whole topic existed, those things were just mysteries.
They were almost like magic. Coordination chemistry connects that abstract theory to real concrete solutions. And historically, once we understood the structure, the applications just exploded. We went from basic discovery to finding EDTA for lead poisoning in the 1940s and then cisplatin for cancer in the 1960s. It's the bridge between what is this and how can we use this?
It really shows the journey of science. Now, I want everyone listening to try a visualization exercise. I think this is great for memory. We're going to visualize a city of chemistry.
Imagine you're looking at a city from high above, maybe from a helicopter. But this isn't a normal city. It's built entirely on coordination chemistry.
Okay, I'm picturing it. The source material codes the buildings by color. So guide us through this map. Where are we looking first?
First, look for the red buildings. These represent biology.
Why red?
Just think of blood. These buildings are the hospitals and our own bodies, and inside them, hemoglobin is carrying oxygen, and enzymes are working nonstop. It's the sector of life.
Okay, red is bio. Now looking at the industrial district, I see some green buildings.
Those are the factories. That represents industry. Inside those green buildings, they are synthesizing ammonia, refining oil, making plastics. They're using coordination catalysts to make those reactions happen efficiently. Green for growth, green for production.
Got it. Red bio, green industry. Now over there, the blue buildings?
These are the medical centers and the pharmacies. This is where you'll find the anti-cancer drugs like cisplatin and the imaging centers that use MRI contrast agents. Blue for healing, blue for medicine.
And finally, on the outskirts, I see some yellow buildings.
Ah, those are the environmental plants, the water treatment facilities. They are working to clean up pollution, removing toxic metals like lead using coordination chemistry.
So we have red, green, blue, yellow. The big takeaway is that this entire city runs on the science of metal-ligand bonding.
It really does. If you remove that one concept, that one type of bonding, the entire city just stops functioning.
That is such a vivid way to remember all the applications. Okay, let's zoom into some of those specific examples now. We started with the blood question. Let's solve that mystery properly. Hemoglobin.
Right, our first observable phenomenon. Hemoglobin is a classic coordination compound. At its very center, it holds an iron ion. specifically iron 2, that's Fe2+.
And what's holding the iron in place?
A huge ligand called a porphyrin ring. You can imagine it like a donut with the iron sitting right in the hole.
Okay, so we have iron sitting in a donut. How does the color change happen?
It's all about what attaches to the iron. When you breathe in, an oxygen molecule, O2, binds to that iron. This changes the entire electronic environment of the metal, and boom, it reflects bright red light. That's your oxygenated blood.
And when it gets to the muscles?
The oxygen detaches. It goes into the tissue where it's needed. The iron is now naked at that spot, or maybe bonded to a water molecule. This shifts the light absorption again, and the color becomes that dark purple or blue.
So breathing is basically just reversible coordination chemistry.
Precisely.
Yeah.
And the key word there is reversible. If that bond were permanent, like what happens with carbon monoxide poisoning, the oxygen would never let go. We wouldn't survive. The coordination bond has to be strong enough to hold, but weak enough to release when needed.
A very delicate balance. Now let's move to the blue building medicine. We touched on a drug called cisplatin.
Yes, cisplatin. This is a huge topic for exams. The formula is PTNH3-2Cl2. That's platinum with 2 ammonias and 2 chlorides attached.
It sounds like a fairly simple molecule.
It is deceptively simple. But here, geometry is destiny. This molecule is square planar. So imagine a square with platinum in the middle. The 2 ammonias are next to each other. That is the cis position.
And that's where the name comes from, cisplatin.
Correct. Now, how does it kill cancer? This molecule goes into a cancer cell and it binds to its DNA. Because of that specific cis shape, it creates a kink in the DNA structure. It basically puts a lock on the DNA so it cannot copy itself.
And if the cancer can't copy its DNA, the cell can't divide.
Exactly. The cell detects this massive damage and it triggers its own death.
You said the geometry is critical. What happens if the ammonias are on opposite sides?
That would be the trans isomer, transplatin. And guess what? It is completely useless as a drug. It just doesn't fit the DNA lock properly.
That is incredible. The same atoms, just arranged differently. And one saves lives while the other does absolutely nothing.
And that is why we stress on isomerism in Unit 5. It's not just about drawing shapes for fun. It is literally the difference between a cure and a useless chemical.
Structure determines function. What a great point. Now let's talk about something a bit scary but fascinating: lead poisoning. Our counterintuitive example from the yellow building.
Yes, this addresses a very serious real-world situation. Suppose a child accidentally swallows paint chips that contain lead.
Okay, so the naive assumption, what I would think is, all right, just flush the system, give them lots of water, and wash it out.
But it doesn't work like that. Lead actually mimics calcium in the body, so it enters the blood, and then it goes and hides in the bones and tissues. It stays there. You can't just wash it out with water.
So how do you send a rescue team in there to get it?
We use a compound called EDTA that stands for ethylenediaminoacetic acid.
That's a mouthful, EDTA.
It is. But forget the name for a second. Just look at the structure. It is a hexadentate ligand.
Break that down for us. Hexa means 6. Dentate must mean teeth.
Exactly. 6 teeth. So think of EDTA as a kind of claw with 6 fingers. When it finds a lead ion, it wraps around it completely, grabbing it from 6 different directions.
Like a cage.
Yes, a chemical cage. This forms an extremely stable complex. The stability constant is very, very high. Once the EDTA grabs the lid, it does not let go.
And then what happens to it?
The body can recognize this EDTA lead cage and safely excrete it in the urine. If we used a weak binder, something with only 1 or 2 fingers, the lid might just break free and cause damage again. It has to be that stable hexadentate grasp to be safe.
That is a brilliant application. We are literally sending in a chemical claw to pull poison out of a person's bones.
It's the perfect example of how stability constants, a concept we study in theory, have a direct life-saving application.
We've covered hemoglobin, cisplatin, and EDTA. These are the titans of this chapter, really. But we need to make sure our listeners are ready for the actual exam paper. Let's do a quick rapid-fire checklist. These are the exam-safe clarity lines. I'll call out the topic. You give me the exam keyword and why it matters.
Let's do it.
Number 1, hemoglobin.
F2+ in a porphyrin ring. The function is reversible binding of oxygen for transport. Remember that word, reversible, it's key.
Number 2, cisplatin.
The formula is PTNH3-2Cl2. The geometry is square planar, and its action is binding to DNA to stop cancer cell division. Important note, only the cis isomer works, not the trans.
Number 3, Enzymes.
They are biocatalysts. Many require metal ions like zinc, copper, or iron coordinated to protein ligands to function. No metal, no catalysis.
Got it. Number 4, EDTA.
Hexadentate ligand. Used in chelation therapy. The key word is high stability constant with toxic metals like lead and mercury, allowing their safe removal.
Number 5, industry.
Catalysts. Coordination complexes are used to facilitate reactions like polymerization or ammonia synthesis.
And finally, number 6, color.
This is due to D-hand-to-D transitions and the specific coordination geometry of the complex. This is the science behind dyes and pigments.
That is a fantastic checklist. If you're listening, maybe just rewind that last minute and write those points down. That's your revision cheat sheet right there.
Absolutely. Those points connect the theory directly to the questions you're going to face in the board exams.
So bringing it all together, we started by just looking at our own wrist, and we've ended up fighting cancer and cleaning up lead poisoning.
It has been quite a journey through this chemical ecosystem.
Let's summarize the key takeaways in a really punchy way for our listeners.
To summarize, first, coordination compounds are the machinery of life. They are not just exam topics. They run the biological and industrial world.
Okay.
Second, think of the Swiss Army knife. One complex can bind or catalyze or produce color, all depending on the ligands you attach to it.
Right.
Third, remember your 3 big examples. Hemoglobin for carrying oxygen, cisplatin for cancer treatment, and EDTA, the 6-fingered claw for removing lead poison.
And the last point.
And finally, structure controls function. Whether it's the square shape of cisplatin or the hexadentate cage of EDTA, the geometry is what solves the problem.
That is a perfect wrap-up. You know, before this deep dive, I just thought of complexes as those things with the confusing square brackets. But now, when I see a green leaf or my own blood or a plastic bottle, I realize there's a coordination compound doing a very specific job right there.
That is the beauty of chemistry, isn't it? It changes your lens. And for the Class 12 student listening, next time you're studying Unit 5, don't just memorize the rules. Look for the applications. When you see a gemstone, think coordination. When you hear about medical treatments, think coordination.
Expert, before we sign off, do you have a final provocative thought for our listener? Something to leave them thinking about?
I will leave you with this thought. Coordination chemistry is incredibly local. It happens at the scale of a single atom and its neighbors, but it is global in its impact. It saves millions of lives and runs billion-dollar industries. It's the perfect example of how the very, very small controls the very, very big.
The very small controls the very big. I love that. Thank you so much for guiding us through the ecosystem of coordination compounds today.
It was my pleasure. Keep studying and keep connecting the dots.
And to our listener, good luck with your studies. You have got this. We'll see you in the next deep dive. Goodbye.